Ever wonder how scientists tell one microbe from another? It's not just about how they look, it's about what they do. In this section of your microbiology lab, you'll go beyond the microscope to explore how microbes metabolize, react, and grow on specialized media. This video will help you connect biochemical tests and selective, differential media to real-world clinical work, because learning how to interpret these tests now will prepare you to make sense of clinical reports later. Microscopy alone will not allow us to identify a microbe to the species level. In this section of your laboratory, you will be introduced to a survey of biochemical tests and selective and differential media used by laboratory professionals to identify microorganisms to their species levels. You will explore a variety of tests, such as the triple sugar iron agar test, catalase test, blood agar, mannitol salt agar, among others. These tests can often prove challenging to understand when first encountered, and therefore often frustrate students. So, let's take a moment to see why an understanding of these tests will help you in your future career as an allied healthcare professional. Tests like the catalase, urease, and triple sugar iron agar TSI tests help identify bacterial species based on metabolic capabilities. These tests are used in clinical microbiology labs to help identify pathogens from patient samples like blood, urine, sputum, or wounds. Identifying the correct bacterial species is key to prescribing effective antibiotics or other treatments. For example, a beta hemolytic reaction on blood agar could suggest a Streptococcus pyogenes infection, which requires specific treatment. These tests also help distinguish harmless microbes from pathogens. For example, a positive coagulase test indicates the presence of Staphylococcus aureus, which is a pathogen known for infections such as abscesses, pneumonia, and sepsis. Other species of staphylococci, like Staphylococcus epidermidis, a member of normal flora of the skin, are coagulase negative. The key takeaway here is that allied health professionals often work alongside clinical laboratory staff or receive reports that include results from these tests. Learning to understand these biochemical tests and selective or differential media in the microbiology lab will help you develop essential diagnostic skills needed to identify pathogens, interpret clinical lab results, and understand how microbes cause disease. Your understanding of what the tests mean and how results are obtained will give you the confidence to communicate with patients about lab findings and help you make sense of lab reports and electronic health records. In this video, you explored the clinical significance of biochemical tests and specialized media in microbial identification. We discussed how tests like catalase, coagulase, TSI, urease, and various agar types help distinguish pathogenic microbes from harmless ones, and how these results are used in patient care. Knowing how to interpret biochemical tests is a stepping stone toward diagnosing disease and recommending the right treatment in real-world healthcare. Microbes reveal their identities in the media. It's like a puzzle, and every test result is a building block. As you observe each color change, bubble, and reaction, you're getting closer to solving who the organism really is.